Pleasure to welcome you all for the fourth day of the GION sponsored workshop on advances in biofuels and bioenergy research. To begin the today's session, I am delighted to welcome Dr. Ramraj Bhupati sir to deliver his presentation on the topic of microbial mining of termite guts for the bioprocessing.
Okay, thank you. Good morning, everyone. Today we are going to talk about termite microbiome and how we could exploit the bacteria in termite guts to various purposes. I also added a wastewater treatment as another topic, so we'll have a little more on discussion on that one. Okay. Um, so if we talk about microbiome, so what is microbiome? Microbiome is the microorganism living on and inside you, all the microorganisms. Okay. So without microorganisms, none of the animals and humans can survive. So we depend on microorganisms. So we have human microbiome. So if you all must have heard about it. So human microbiome we carry within us 3 times more bacteria than our own human cell. Okay, so humans are made up of 1 trillion human cells. If you only count how many bacteria inside you, you have 100 trillion human cells. So I mean humans have 33 trillion, so we have 100 trillion bacteria. So we have 3 times more bacteria inside us than our own human cells. Okay. So same thing is true for all other animals, whether it's a cow, whether it is an insect, they all depend on microorganism for digestion. So if you want to live a healthy life, you want to take care of your microbiome, the bacteria in your guts, in your intestine. So they are the one doing the job. Okay. So anything that you do depends on microorganism in your GI tract, in your guts. Okay, so like you are stressed out, your mood is swinging back and forth. It's because of microorganism. Believe it or not. Okay, so they are your second brain. Whatever activity they have biochemically, they produce enzymes. The enzymes convey the message through vagus nerve that connected to the brain. So everything that you do is the activity of microorganisms in your gut. So that's why you need to take care of your digestive health to have a better health as a whole. Okay, so that's why nowadays there is a lot of food industry now come up with probiotic, prebiotic, all that stuff become commercialized because of this research about human microbiome. So we are discovering more about good bacteria that outlive. So one more fact about if you have a digestive issue like we call irritable bowel syndrome, which people have problem, digesting food. So because they lost the diversity of microorganisms in the gut, they only have 1 or 2 species dominating. So we're supposed to have 2,000 different species inside, and there are like trillions of them inside you. So those people lost the diversity. They are the ones that have trouble in holding food and all that stuff. So let's just tell you a story about microbiome. So same thing is true in insect. In this case, termites. So termite eat wood, right? Wood is basically same topic we talked about: lignin, cellulose, hemicellulose. Okay, termite cannot digest those at all. They eat them. Okay, but who is digesting those? The microbiome inside the termite. Okay, so if you eliminate those microbiome, termite will die. Okay, if you want to kill the termite, kill the microbes. Okay, so that's the story here. So we did some microbial analysis of termite guts. What do they do? What most of the group of microorganisms carry out? What kind of function? That's what I'm going to talk about today. So termites is really bad because they do damage. In the U.S., they cause $1 billion damage per year, eating infrastructure. One of the major case that happened in New Orleans, where I live, we had the Hurricane Katrina in 2005, and that time the whole city of New Orleans was underwater for 2 weeks, right? It's because The New Orleans is levied by big levee system, and the reason that they got flooded was termite damage. The termite eat that levee and weakened the levee. When hurricane hit, there was no protection. It just levee broke and water came in and kind of flooded the city. It was a big mess for 2 weeks. Water couldn't get out. So that's an example of termite cause economic damage. Okay. Termite also in nature play a good role. They are efficient nutrient cyclers. They cycle nutrient on a global scale. So they have a key role in ecosystem. They carry out carbon turnover, basically carbon cycle. They really play a key role. They have their own role in ecosystem. At the same time, they also cause economic damage if you in the city, like big cities. So where I come from, there are two kinds of termite. As one is the invasive species that's called Formosan termite. That happened to come from China, from the timber they imported for the railway track. So that got escaped and colonized and become very nuisance there. But we have a native termite. The difference between the invasive, this is the invasive one, Formosan termite, and the native termite, this head has a teardrop shape. That's how we can identify whether it is a native or invasive species. Okay. So both termites are really damaging the city infrastructure. And as you know, these are social insects. They have the hierarchy. We have a queen. We have soldiers and workers. They all have different function in their colonies. Okay, so most of the work is carried out by the worker termite. Okay. So coming back to the topic of discussion about bioenergy, if you look at the biomass, the same story we've been talking about last few days. It's wood. Contains the three major polymer: lignin, cellulose, hemicellulose, and cellulose is made up of 100% glucose, and the glucose is put together by the alpha 1,4 bond. So that's why it's a polysaccharide. And if you break the alpha 1,4 bond, you release glucose 100%. Okay, so you need cellulase enzyme to break cellulose into glucose. Okay. And hemicellulose is made up of a lot of different sugars. These are the pentoses and hexoses. These are the sugars that put together hemicellulose polymers. Okay, and the different bonds in there, and you break the bond, you release the sugar. So when we talk about lignocellulosic ethanol production, we're talking about biomass to get these two sugars, cellulose and hemicellulose, release glucose and xylose 6 sugar. Okay. And then the third component is lignin. Okay, the lignin is holding that cellulose, hemicellulose together. So that's why you have pre-treatment, enzymatic saccharification to get the sugar out. Right. All of this is happening inside termite guts, and termite guts is a mini bioreactor. Okay, so there's so many different species of microorganisms carry out breaking down the wood biomass that termite eat, and that releases energy-rich acetic acid that termite use for an energy source. Okay, so 100% termite depend on microorganisms for digestion of wood. Okay, so phenol, when you break down phenol, what do you get? When you break down lignin, what do you get? You get phenol. And most of the phenols are these three compounds we talked about over and over again. Those are the coniferal alcohol, coumaryl alcohol, synapyl alcohol. So these are put together as in a huge polyphenolic compound that we call lignin. Okay. So when you break this lignin down, it's done by microorganism inside termite guts, you get phenol. Okay. And phenol can be used by bacteria as carbon source. So this is a small picture of what kind of organisms in the termite. There are 100 times more diversity there. I break it down for you to just to keep the subject going here. So termite would eat the wood. The termite is eating the wood. The major organism that break a lignin is not bacteria. It is these protozoan. Okay? The flagellated protozoan are the one that break down lignin, release cellulose, hemicellulose. Okay, and the same thing, these bacteria also, these protozoan also break down some of those into acetate. Okay, so this is the key player in termite gut. If they don't have this protozoa, they cannot release cellulose, hemicellulose from the woody biomass termite is eating. Okay, so these are the key protozoan that break the lignin off the biomass, get the cellulose and hemicellulose out. Then we have other bacteria that secrete enzyme that break down cellulose into sugar and hemicellulose into xylosic sugar. The key intermediate is they produce exactly like in an anaerobic digester. They produce acetate, hydrogen and CO2. Okay? And acetate is used by termite as energy source, but also we have methanogenic bacteria in termite, and they does the same function like as we discussed in anaerobic digestion of taking these compounds to make methane. So termite produce methane. One of those largest methane contributor is apart from cows, termite also contribute methane. Okay. So this is a picture of what's happening inside termite guts. There's a lot of interaction of different groups of microorganisms that carry out digestion of wood material that termite eat. So termite is like a bioreactor. They just bring in the feedstock. The feedstock is wood they eat, and it causes damage to the infrastructure, but they all depend on these organisms to get energy by breaking it down. Okay? So our main goal when I got the research project is to isolate these protozoa from the termite and grow them in large quantity in bioreactor and get the enzyme, what kind of enzyme they do to break the lignin. Okay? Instead of going chemical pretreatment, we couldn't go in the biological route. But I was not successful in isolating this flagellated protozoa. It's very, very hard to mimic the conditions of termite gut in a test tube. And we were not successful. We tried so many different media, nothing worked. So, just that was our main goal. But we did other work, and that's what I'm presenting today. So yeah, the objective is, if you look at termite, they eat all kinds of wood. Okay, in nature there are so many different kinds of tree, and so we wanted to find out if the diet of the termite make a difference in bacterial colony. Okay, so we went out and looked at different termite colonies that are living close to different tree, and picked termite, brought to the lab, pull out the guts, squeeze the juice into the media, and looked at different carbon source, what they are trying to do. So, okay? So these are the main objective of a project. Determine the microbiome structure of 4 different termite colonies, determine the relationship between the wood type and number of bacteria capable of utilizing common carbon source in the termite gut, determine the efficiency of phenol degradation by, we isolated successfully one species called Acinetobacter, and break down and look at the metabolic pathway of how it breaks the phenol into carbon dioxide. So let's look at the first objective. So my student went out, collected termite from different sources. We picked 10 worker termite from a colony and brought to the lab, surface sterilized the termite with alcohol wipe, and homogenized and pulled the termite guts and did the 16S and 18S sequence and look at the biodiversity of microorganisms. Okay, and they compare the results. That's our first objective. What kind of microbes are living in these different termite colony eating different wood types. Okay, so just to orient where the sample come from. So we picked one in this Bioprocessing with the Tupelo is a tree they are eating, and then we have St. Francisville, that's the oak tree, and we have 4 colonies, 2 from Bioprocessing, both are from Tupelo tree, and 2 from St. Francisville. One was eating maple, another was eating oak. Okay, So this is New Orleans, the big cities, and that's the capital of the state, Baton Rouge. Okay. So first we looked at the protozoa. That's the key. Protozoa break down the lignin out of the wood. So if you look at these different sources of termite, these are eating different tree types and where the sample come from. They all have one common termite in all of them. This Trichometopsis, all of them have the same kind of protozoa. And also they also have the second group, they all have it. The third group varies. These Tupelo 2 didn't have the Trichomonas, and then these oak eating and maple eating has additional protozoa, different species. Okay, so this tells us the diet matters. Okay, so what kind of wood they are eating, particular type of protozoa dominating there, because maybe oak and maple are hard to break it down. They need more kinds of protozoa, different species to work in together. Okay, so that tells us the diet matters, and same thing is true for us. Our microbiome is different depending on what kind of food you're eating. If you are a meat eater or a vegetarian, your microbiome is totally different. Okay. So then we looked at the bacterial diversity. Okay. So again, you look at the bacterial diversity. We have again same termite that from Tupelo eating Bioprocessing and Tupelo eating Bioprocessing second location, and in St. Francisville oak eating maple eating. If you look at this pie chart, all of these colonies have these group of bacteria present. The diversity is there, but it is same bacteria, no different. But what is different is the percentage is different. You look at the dark blue color. The dark blue color is our Bacteroides. Okay, the Bacteroides is in this particular colony is more than 40%, whereas here is 8%, is 16%, and here is 13%. But every one of these termites have all this group of bacteria. There is no difference in species, but the species dominance is different. That's because of the diet. Okay, so that again tells us. Feedstock is, we talked about variation feedstock, right? Different feedstock are different percentage of hemicellulose, cellulose, lignin. Exactly that's what's telling here. Okay, so the bacteria colony and the species dominance matter. That's because of your feedstock is different. So the organism inside termite is adapting to that. So number of population in one species is high, depending on what type of wood they are eating, and another species is different because it's softwood, hardwood, different type of wood they are coming out. Okay, so if you look at the name, these are the thing that major bacterial group we identified using our molecular work. Every colonies we looked at, they all have the same species, but the percentage varies in these termites. And then we further analyze phyla, class, order, family. We went all the way, but we looked at this location, and you can see total phyla in each group, total class, total order, total families. Okay, but what is the key here is the uniqueness. When I say uniqueness, is that when I say one unique phyla found in this termite means you didn't find the same phyla in any other wood type. Okay, so as you look at all the way in family, and you can see more uniqueness you are finding. Okay, same thing in each wood type. Okay, but you have some of them are same phyla, some of them say, but the uniqueness varies from wood type to wood type. Okay, so that again tells us they are tailor-made. They are tinkering with their biodiversity depending on the feedstock. The feedstock is different, so the bacteria, the termite colonies have unique family, unique species in different colonies. Okay, so this is just teaching us the bacterial evolution and termite termite is our basically conveyor belt, and it gives feeding the bacteria, and whatever wood they are eating, that termite colony, bacterial colony inside the termite is adapting and changing. The structure of the diversity is different. The species uniqueness is different, and that's what's telling us by looking at this basic microbiology inside the guts. Okay. So the story about termite is when termites are hatched out of the egg and developed into adult termite, they are sterile inside. The termite colony has absolutely no bacteria. They get the bacteria like feeding on feces in the colony. They enrich themselves with different microorganisms. Okay, the termite microbial diversity depends on the founders of the colony, because termites swarm every few months. The queen leaves and establish a colony in a different place. So the microbiome is coming from the swarmers that go establish a new territory, a new colony. Okay, so whatever diversity the queen and the soldier that met with the queen have, that is the founder microbiome. That founder microbiome is going to be unique, and whatever termite they are producing in the new place will have that uniqueness in there. Okay, so that is the story here. So every time termite moves and establish, whoever found that colony, whatever bacterial diversity they have will be what is in that colony. Okay, so the founder carries the, in simple term, inoculum, right? When you inoculate bacteria, you grow bacteria. So basically the inoculum is coming from the founders for the whole colony. Okay. Another story is neighboring colonies do not interact. When there are two colonies, living close to each other, you don't see microbial diversity. They are exchanging microorganisms. They just separate, completely separate. They do not interact with among each other. So some of the functionality of the group, there's a lot more. I simplified it. There we have acetogenic organisms that produce acetate, which is important for our biofuel thing. So a couple of them, protozoan also produces a lot of spirochetes and tropomona bacteria are there. We also found a lot of nitrogen fixing bacteria, because termite diet is nitrogen poor. Termite doesn't have a lot of nitrogen in the wood. Okay, so we found a lot of bacteria that can fix nitrogen from the air, convert them to ammonia. They are nitrogen fixing bacteria. So these bacteria play a key role, provide nitrogen source to the guts and the termite. Okay, so we went through a different group and what they do, but I'm just showing you two key examples. So the diet is nitrogen poor. There's no nitrogen in the wood. So bacteria is fulfilling that role, providing nitrogen by fixing free nitrogen from atmospheric gas. Okay, so atmospheric gas are close to 80% nitrogen, right? So bacteria fix that. The second objective is to find out the relationship between wood type and number of bacteria capable of utilizing carbon source in termite guts. So now what we did was we pulled out from different colonies. We didn't do pure culture. We just pulled the juice of the guts, put them in the media with different carbon source, cellobiose, phenol, glucose, and see, are there any difference from colony to colony? That's what we are trying to look at. So if you look at these wood type, we have oak, we have tupelo, we have maple. So the termite basically eating all these wood type. The microbiome inside the termite is converting the wood to different intermediate. Okay, the cellulose broken down to cellobiose, lignin broken down to phenol, and all the other major polymeric compounds converted to acetate. That's the energy source for termite, the energy source for other bacteria like methanogens in the inside. Okay, so this is what's happening. So we want to find out these metabolites that's produced inside, are they different? Are they same? Are the bacteria going to show dominant preference to one carbon source over the other? Okay. So this is my grad student pulling out termite guts aseptically. We took 10 workers per colony, alcohol wash, abdomen removed and homogenized, did serial dilution and did colony plate counts with media contains only specific carbon source, phenol, acetate, cellobiose. Okay. Yeah, that's a method, and these are the carbon source we tried, and we put a control which is a nutrient rich biolog media, and then we grow and count the colonies and report the result and did the statistics on it. Okay. So that's the result. You can see different wood eating termite colonies, maple, tupelo, oak, and they're grown on phenol and nutrient rich biolog media, cellobiose, acetate. The beauty of it is, but no matter where they come from, they all grow in all the metabolites. There is no preference at all. There's a minor statistical significance in case of phenol. Phenol preference is different from colony to colony, but all other metabolites is almost the same. Okay, where they come from, what wood type they eat, the bacteria, break it down. All the carbon source we gave. That makes sense because these are the major metabolites that's inside the termite gut. So bacteria is using that and break it down further. Okay, so that tells us no matter what type of wood type, the metabolites is the same. Those are the three major ones: acetate, cellobiose, and phenol. Okay, and then termite gut. If you look at it, the periphery of the termite is aerobic. When you go little inside, facultative. When you go right into the middle of the gut, it's completely anaerobic. It's exactly like your anaerobic digester. The top of the digester is a little bit aerobic, and then it will go down a little bit, it's facultative, and then the bottom is anaerobic, right? So exactly same thing in your termite gut. So what we did was we did the same experiment and grow one anaerobically, one aerobically, and count how many bacteria are there. Are there any difference? Okay. To our surprise, there is no difference. Statistically, there is no difference. We found equal number of aerobic bacteria and equal number of anaerobic bacteria, no matter what colony it comes from. Okay, whether it is a wood oak eating colony or in a natal colony. Okay, so that's something that equal amount of aerobes and equal amount of anaerobes in the termite guts. And then we looked at the diet. Okay, so we pulled all the data together, where they come from. Are they statistically different? We found there is no statistically different. So they all does the same function, whether it's the termite colony come from maple or oak, it doesn't matter. The metabolite is the same, so there is no difference in bacterial dominance of one over the other in carbon preference. Okay. The same thing, that the carbon preference is the same, no statistical difference at all. Then the third objective is to look at phenol degradation by pure culture. So original goal was to isolate protozoa, try to find lignin degrading enzyme. We were not successful, but we isolate other pure cultures, and one of the pure culture is this Acinetobacter tenduii, and this, through our research, we found this bacteria play a key role in survival of termite. Why? Because when you break down lignin, what is the major metabolite you get? Phenol, right? Phenol is a known antimicrobial compound. Phenol will kill microbes, right? So what this particular bacteria is doing is it breaks the phenol down, so keep the toxicity level low. so all the other microorganisms can survive and do the job. Okay? So this particular bacteria has a role to play inside the termite gut, keeping that phenol concentration low. Okay? That's what we found out in this research. Okay? So we looked at this particular species. We did more research on that. I'm going to share the data with you. So the bacteria degraded phenol completely. We looked at two species from this termite gut. One was Acinetobacter tanduii, other was Acinetobacter berzynii, and both of them are obligate aerobic bacteria, the aromatic degrading bacteria. You can find this Acinetobacter in sewage and industrial effluent because they are aromatic degrading bacteria. We identified it using biolog and molecular method, and we grow them in culture media using different carbon source, phenol and glucose and co-metabolic combining phenol glucose together. And to our surprise, this particular species cannot metabolize glucose. That was a very surprising result because every bacteria grow on glucose. Okay, so this bacteria prefer phenol. Okay, and we did the statistics on it, the growth after we growing this in different carbon source. So here's the result. So this Acinetobacter tanduii in the literature, when you look at it, they eat glucose, no problem. This particular one from termite gut lost its gene to metabolize glucose because there is no glucose in termite gut, so there is no use for it for this bacteria. So it evolved into sophisticated, only phenol degrading efficiency it improved. So it completely eliminated the gene that responsible for glucose metabolism. So you look at the result, the phenol grown culture beautifully grew, no problem. Glucose grown culture did not grow at all. Very weak growth, but statistically big difference. That's a p-value. Okay? When you put phenol and glucose together as a co-substrate, there's a long lag. So what it indicates is glucose is suppressing the activity of bacteria. The bacteria has to overcome glucose, is somehow inhibiting phenolic enzyme expression. But after a while, it's doing the job of secreting the enzyme, and you can see the growth coming back. So that's is the evolutionary role of how microorganisms adapting to conditions they are living in. Okay, so this is one of the things that we are totally shocked as the bacteria. Other Acinetobacter we collect from culture collection grow beautifully in glucose, and this one did not, and they prefer phenol because they've been phenol degrading is their role inside termite guts. Okay, and just to see carbon removal, we did the COD analysis. The same story. Percent removal of COD was really high in phenol grown culture. In glucose grown culture was not much, statistically big difference, that's a p-value. And when you have the co-metabolic conditions, it took a while, then it removed 40 to 50% of carbon. Okay, mostly it's phenolic carbon, glucose carbon is still there. Okay, so that tells us the story that this particular bacteria is very, 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 very specific and has a key role to decrease the toxicity of phenol and keep the other bacteria in the termite guts alive, so they can do their job of keeping termite alive. Okay, so if you want to kill the termite, you kill the Acinetobacter. Okay, you kill the Acinetobacter, then phenol will accumulate. Phenol will kill all other microbes, and termite will die. Okay? So the last objective on this particular research is we construct a metabolic pathway, how phenol is completely breaking down. So we grew culture again, repeat the experiment. We compared with the pure culture and then compared with the consortium, okay, without pure culture. We grew it, and then this is the experimental setup. And same story, it did not grow when there was no, this is percent removal, we're not showing the growth. The percent removal was high when in phenol grown culture compared to glucose grown culture. and then co-metabolic is small amount was removed. But when you switch to the consortium, the whole gut pulled out, the story flipped. Okay, you see the glucose grown culture removed more carbon than phenol grown. But here there is, because consortium has a lot of different bacteria, they all prefer glucose, whereas pure culture, this Acinetobacter only prefer phenol. That makes sense, right? So that tells us that this particular bacteria has a unique role in the ecosystem of termite guts. Okay. Then we did the GCMS of the culture, and you can see these are the metabolites from phenol. We found catechol, cis-mecanic acid, succinic acid, oxaloacetic acid. The final product was acetic acid. Okay. This acetic acid is what gives the energy source for termite, energy source for other organisms inside termite. So phenol is broken down to acetic acid. The difference here is the phenol grown culture and phenol and glucose grown culture. The phenol and glucose grown culture lost its ability to make a lot of acetic acid. The pathway shut down right here because it cannot metabolize glucose. Everything is accumulating here. Whereas when you only provide phenol as carbon source, it goes everything all metabolized and everything go to acetic acid. Okay. So that's why the construction of pathway looks like this. So phenol is broken down to catechol, cis-mecanic acid, then you have cis-mecanate and 3-ketoadipate and ketoadipate CoA, and then we have the final product acetyl CoA, and then we have succinate. So what happens when you have glucose? These things are shut down. It's oxaloacetate, succinates are accumulating in the system. Okay, so the bacteria lost the ability of glycolysis. You need glycolysis to break down glucose, right? So the bacteria lost that gene, and so they cannot do it. The first step in metabolism of glucose. So Acinetobacter is incapable of glycolysis. So that's why glucose grown culture did not grow, as well as phenol grown culture. Okay. So, and then when you have the two things together, glucose is the suppressor. It's suppressing the activity of bacteria. So we are accumulating more oxaloacetate and succinate. We are not going into acetyl CoA to go to TCA cycle. So acetyl CoA is our input into your Krebs cycle, right? So everything is accumulating right here when you have phenol and glucose together. Okay, so this biochemical pathway is telling a story that there's one particular bacteria in this termite gut that can take the lignin metabolite phenol and break it down to acetate, and that has a key role in termite survival because phenol is a known antimicrobial. So it keeps the phenol concentration down in the termite gut so other microbes can survive, do the job, and keep the termite alive. Okay, so that's the story of this particular research. So we compare the other Acinetobacter. Other Acinetobacter prefer glucose, and they are known aromatic degrading bacteria. Isolated from other sources have a totally different biochemical property than the one that we did research here. Okay. Again, emphasize a point here. This bacteria has a key role in removing phenol toxicity. Okay. Then what we did was, okay, how high phenol concentration this particular bacteria will take it. So we started out stepwise. We grew them at 300 milligram per liter, 400, 500, 600, 700 milligram in a culture bottle. So what we did was first we grew on 300 milligram per liter, no problem, they grow really high. Then we took a culture from here as an inoculum and inoculated into 400 milligram per liter media, and no problem they grew. And then we took a culture from the 400 milligram adapted culture and provide 500 milligram. It took a lag period. The 500 milligram took a little toxic to them, it's killing the bacteria, but few of the cells survive and they adapted and they grow. And then we took a culture from this and then gave a higher dose. It took a long lag, and most of them died, few cells survived. I mean, they adapted to the high concentration and they multiplied and they started growing on that. That culture went up. Okay. That's the maximum it can tolerate. When it went to 700 milligram, it just everything died. So this is one of the highest phenol concentration reported in the literature. It is called directed evolution. We are directing evolutionary survival of bacteria by repeating the experiment. By bacteria that survived at higher dose, we subculture them with even higher dose in this experiment. Step by step, we increase more concentration until it reached the critical point where everything died. Okay? The limit here is 600, 700 milligram. None of them survived. Okay? And then we wanted to see phenol is a phenol, right? Then there are substituted nitroaromatic. There could be nitrophenol, chlorophenol, So we want to find out whether all those other solvent industrial chemicals that we use that are like explosive compound, they are all basically nitroaromatic compound, whether these bacteria are going to break down the substituted phenol. So we grow this bacteria in these different chemical. Toluene has a methyl group, right? And nitrophenol has nitro group in a phenol compound. Dinitrophenol has a 2 nitro group, and tri has 3. These bacteria grew in all of this, with the nitro substituted compound and chloro substituted compound. It doesn't matter, it just break it down everything and use a sole carbon source. And then we also did the degradation. Not only we showing it grew, it also removed the concentration of all these substituted compound. Okay? And then we could adapt this bacteria in paper and pulp mill industry, which is all full of phenolic compound in paper and pulp mill. So this could be easily used as a bioremediating bacteria for paper and pulp industry, which is basically a lot of phenolic compound in its constituent. The summary of this research is the diet may affect relative abundance of bacteria, but the species of bacteria is the same. What type of wood they are eating, it doesn't matter. Species remains the same, but the abundance varies. Okay? The distance colonies are unique. Each one has a unique group of phylum, family, order, species. There is some unique bacteria present in termite guts, depending on what type of wood they are eating. Okay. The preferred substrate is cellobiose. That makes sense because cellulose, when you break down, you get cellobiose before you get glucose, right? So cellobiose is the preferred substrate for all the consortium come from various termite colonies. Okay. And then one particular pure culture we worked on could be used for bioremediating agent for phenolic compounds. Okay. At this time, I'm going to switch gear. I'm going to talk about wastewater treatment, in particular industry, in this case shrimp aquaculture industry, okay, in a sustainable way. So when we use the wastewater system treatment, we want to make sure we are completely taking care of everything that comes out in the industry. We're not just only treating the waste, we want to make sure the solids taken care of and absolutely everything is completely treated. So I just want to share with you the rest of the time I have this unique story, but it's totally different from biofuel, but there's more about sustainability. Okay. So the story is about aquaculture, particularly shrimp aquaculture. Okay. So the aquaculture industry is a big industry worldwide. All the fisheries we eat mostly come from aquaculture. The wild caught fishery is going down, right? We are depleting fish in the ocean. Most of them are aquaculture fish. That's what we are eating. So if you look at this graph, so the aquaculture industry is keep going up, All right, that's the aquaculture industry. Wild caught fish is going down. Okay, so our protein source, if you are a fish eating person, mostly coming from aquaculture industry. Okay. So if you look at the production of aquaculture, countries that are dominating it, China, and most of them are Asian countries. The aquaculture is booming in Asia. Okay, in terms of quantity of aquaculture fish and different species they're growing, also dollar value. Okay, so mostly China and the other Asian countries. If you look at U.S., very small amount. All right. The reason for that is environmental regulation. Environmental regulation in the U.S. is so stringent, very tough. You have to treat the wastewater. You cannot let it out. And if you're growing marine aquaculture, it has to be 50 miles away from the coast. You cannot have marine aquaculture close to the ocean because of security of releasing your disease species into the ocean and creating problem in the ocean. So they have a very stringent regulation. So the major cost in the U.S. for aquaculture is to shipping marine water inland, large quantity of them if you want to grow fish, marine fish especially. Or you have to dig deep well, you get the brine water coming out of the ground. Okay, so that's a major cost in the shrimp industry in the U.S. because of environmental regulation. The other countries are a little lax regulation. I don't know. That's why they are dominating compared to U.S. And among all species, the shrimp is the dominant one in terms of production and dollar value. Shrimp has more dollar economic importance of aquaculture. And then if you look at the shrimp worldwide, most of them, three-fourths of them is aquaculture shrimp. Very small amount is wild caught. Okay. And then the economics of shrimp, again dominant by China and Asian countries, very little from North America and the U.S. I'm going backwards. So if you look at the species among the fisheries, crustaceans are the one dominating, okay, like shrimp and crab. Those have high dollar value. And again, China is the dominant player, and you can break it down. India is somewhere here, 7% Indian shrimp production worldwide. And you can see China, Thailand, Indonesia, Vietnam, India. Okay. Look at the U.S., very small amount. Okay. The reason for that is again environmental regulation, putting the farmers out of business. They cannot compete with the other country. Okay. The particular shrimp that really preferred for aquaculture is called Pacific white shrimp. Scientifically, it is called Litopanaeus vaname is the name of the shrimp. And then we have second one is the monodon. Okay, because of this regulation of biosecurity, the U.S. Farmers have tough time competing. You can see the industry going down. Shrimp production in the U.S. really going down now. It's small amount of farmers left in the U.S. This caused problem in the U.S. because believe it or not, the trade deficit Trump is now bragging about is shrimp is the major one. The U.S. guys eat a lot of shrimp, so they are the second largest trade deficit product. They import so much of them from Asian countries. Okay, so this caused a big seafood deficit, trade deficit in the U.S. Okay, it's become a big economic issues. So the USDA, United States Department of Agriculture, formed a consortium, is called U.S. Marine Shrimp Farming Consortium. Our university is part of them. Okay, so Nickel State, which I am the PI, and then all the other institutes are doing research to help these farmers. So somehow we can improve trade deficit. That's a tough order to do that. So we are doing our role as researchers to help the country. So our objective is to increase domestic shrimp production, to become competitive in the world shrimp aquaculture, and develop advanced farming technologies through science, so we can improve the yield. Okay, so each institute has a role. Some of them are disease pathologists, some of them are virologists, some of them are breeders, some of the genetics. They want to have a good quality shrimp, disease resistant shrimp. But our institute, I am the wastewater guy, so I take care of the wastewater coming out of the shrimp industry. So environmental issue is coastal pond culture is a no-no. In the US it has to be 50 miles inland. They're worried about the salt water getting into the seawater exchange. And then we have pathogen introduction, waste and chemical into the ocean, low feed conversion ratio. These are a major problem. That's why the US put a stringent rule. You have to have marine shrimp away from the coast. Okay? So trucking seawater is a major expense. So that's why we come up with recirculating raceways. You bring one-time seawater, that's it. We're going to constantly reusing them over and over and over. That's through wastewater treatment. You want to treat wastewater, keep your salt water run as much many times as aquaculture you can produce. Okay, so that was a burden put on me. Okay, so I got to come up with a solution. So we want to create a system that has little or no water exchange at all. We have to have a biosecure facility, higher infrastructure cost. These are some of the problems that we have to overcome. Okay. So if you look at the shrimp waste, it's a really high protein diet they feed the shrimp. 40% of the diet they feed, this is the pellet they feed, this is 40% is protein. So the protein rich feed cause ammonia production. Okay, when protein is broken down, you get ammonia. Ammonia is toxic to the shrimp. So when you do your wastewater, you need to remove the ammonia, otherwise you cannot recirculate the water. You're going to kill the next batch of shrimp. Okay, so this is how it looks like in the first batch of shrimp aquaculture. That's the sludge coming out, and ammonia is so high, and then we had to preserve all the salt in it, so we can recirculate the salt, so we don't have to truck seawater again. So these are the problem that we had to do over science to overcome that. Okay. So this is the key target. So we want to bring the ammonia to 3.55 at 25 parts per thousand salinity, and nitrate to 15.2, nitrate to 158. Anything more than that, shrimp will die. So we in the wastewater that we're trying to treat to bring this level to the tolerable level for shrimp. Okay. So we looked at it. We looked at, okay, nature has a way to do it, so why we need to reinvent the wheel? So we looked at nitrogen cycle. So let's mimic nitrogen cycle. Okay. In nitrogen cycle, we have operate your wastewater in two cycles, aerobic and anaerobic. Aerobic mode, you convert ammonia to nitrate. Ammonia is oxidized to nitrite and nitrate. And then you switch to anaerobic mode, you denitrify nitrate back to nitrogen gas. That's what this nitrogen cycle is in the ocean. So we wanted to mimic this same thing, the nitrification and denitrification, we wanted to do that. Okay, so nitrification is converting ammonia to nitrate, denitrification is converting nitrate to nitrogen gas. Okay, so we want to repeat that in our system. So we created a reactor called sequencing batch reactor. You can operate them in the batch, and you switch to aerobic and anaerobic mode to convert the ammonia to nitrogen gas, and it needs carbon source. We preferred molasses, which is inexpensive, coming from sugar industry, to add molasses in a denitrification stage. So this is how a SBR look like. Sequencing batch reactor, you fill them in batch of wastewater, and then you operate aerobically to do your nitrification reaction, switch to anaerobically to a denitrification, go back to aerobic to remove some more carbon, leftover carbon, and then you settle, decant, you get the water back into the shrimp production and deal with the sludge separately, and then until the new batch come in the bioreactor, the wastewater reactor sits idle, and when new batch comes in, you fill back again. That's why it's called sequencing batch reactor. Okay? So and then we had to take care of biosolid. The biosolids that accumulate inside has a lot of salt. Try to recover the salt, and then find a use for the biosolid. How we can best use in a sustainable way. Okay? So that's the objective, to exploit the nitrogen cycle, build a SPR, and look at the recycling and recycle the water, treated water back to shrimp production, and take care of the biosolid as a fertilizer, and look for what kind of microbes are there in the SPR system doing the job. So this is just a simple biochemistry of nitrogen cycle. So during aerobic process, ammonia is converted to nitrate, that's your nitrification. The denitrification is an anaerobic process. Nitrate is going to nitrite, nitrous oxide, and nitrogen gas back into the atmosphere. Okay? So you operate part of the time aerobically, part of the time anaerobically. Okay? So we started a small bucket reactor. We need to figure out the sequence, how long you need to be aerobic, how long you have to be anaerobic. So you play around in the lab bioreactor. And this is the parameters we analyzed, and these are the farmers that produce shrimp, and we got the waste from this bead filter. That's where the water backwash go into circulation. We collect the wastewater there and analyzed, and you can see this is where the problem is. You need to bring it to 3.5 to 15 and 150. Okay, these are really high. We cannot circulate the water back into the production unless these concentrations come down. So we did an experiment. We played around. This is aerobic mode. This is anaerobic mode. Aerobic mode, you can see the COD going down in the anaerobic mode versus aerobic mode. Aerobic mode is an autotroph. They don't use carbon. Anaerobic mode is a heterotroph. They break down the carbon. And then we let's show the soluble oxygen demand, aerobic, anaerobic, aerobic, anaerobic, aerobic. We want to play around the time, see how long it takes for each sequence. And then we'll look at the ammonia concentration. So when you operate aerobically, the ammonia came down, which is all converted to nitrate, right? And so when you switch anaerobically, the nitrate is removed to nitrogen gas. And then this is nitrite nitrogen. You can see initially there was less nitrate, but all the ammonia converted to nitrate at the end of the aerobic stage. Okay? When you switch to anaerobic, the nitrate came down. Okay? Exactly what the nitrogen cycle is doing. We are mimicking that in a small scale. Okay? And then nitrate, same story. Nitrate accumulates after aerobic stage because the nitrification ammonia is converted to nitrate. When you switch to anaerobic, nitrate coming down. We did in cycles, several cycles. Then what happens is nitrate accumulates. It doesn't go down because the carbon in the system is less. When there is no carbon, it shuts down. So you have to add some carbon. So that's why denitrification, you need to adjust how much carbon it needs to completely remove the nitrate. So we added a small dose of molasses, all the nitrate come down to safe level. Okay? So this is how we played around with a small scale bioreactor. And then it also removed the sludge. That's a good thing. We don't want too much sludge in our system. This bioreactor removed 20% of sludge. Bacteria took care of the sludge digestion. And then we wanted to play with the temperature salinity, because if you look at farm shrimp farm in the U.S. Spread out in different states, and they all different temperature. The southern states high temperature, the northern state cold conditions, and the salinity level varies because you can grow shrimp from 5 parts per thousand to 30, 40 parts per thousand. Okay, so we wanted to see whether the bacteria that we are developing can adapt to this wide range of temperature, a wide range of salinity. Okay, so that's what this experiment is. Okay, so we did again the same thing, brought the waste and did the analysis, again high in ammonia, nitrate and nitrite, and then play around in a small bioreactor. We varied different temperature, different state, 22, 28, 37, 40 degree, operate the reactor in different, we had a 2 days aerobic, 3 days anaerobic, went back to aerobic to polishing step, settle, and 1 day. So it's a 1 week reaction. The whole operation is 1 week. That's the sequence we adapted from our previous experiment. And you can see the result. So different temperature, 22, 28, 37, 45. Aerobic mode, ammonia came down beautifully, no problem. Okay. All the temperature worked really well. The one that really struggled was 45. When you increase the temperature, the efficiency went down. Wide temperature range, we can operate without any trouble. And then nitrate level is the same thing. No problem with whatever temperature we use. The nitrate went down when you switch to anaerobic mode. Whatever nitrate accumulate went down except this 45 degrees. And then when you did the nitrate, the same story, all temperature we operated worked really well, removed all the nitrate except the high temperature. Okay. Same story with carbon removal, the high temperature didn't like it, but the rest of the temperature we tested worked really well. Then we switched to salinity because different state produce different salinity. The closer to your ocean your state is, you can have high salinity. We have inland shrimp production, they mix with fresh water and go low salinity as much as the shrimp can take. So here we tested based on different state farming experience. So here we have salinity of 5, 15, 28, 40 parts per thousand sodium chloride, and look at whether our bacteria can adapt to this wide range of salinity. Okay? So we kept operation temperature to 22 degrees, one temperature, but we vary our operation in our bioreactor to different salinity level. All the salinity worked in ammonia removal, no problem, aerobic, anaerobic mode, except the low salt, the 5 parts per thousand, the salinity level did not work. Okay, as efficient as others because these bacteria that we started out with 25 parts per thousand adapted to that salinity, but it adapted to other salinity. Okay, but when the salinity to more close to the freshwater level at 5 parts per thousand, the efficiency went down. And then we looked at the same experiment. We looked at the nitrate concentration. So ammonia converted to nitrate during aerobic operation, anaerobic operation, they all came down except 5 parts per thousand. Okay? And then we switched to nitrate in the same bioreactor, NO3. Aerobic it accumulated, anaerobic it went down, denitrification, 5 parts per thousand did not do well. Same story with COD. So that tells us the extreme conditions did not work, 40 degree centigrade and 5 parts per thousand did not work. The rest of the conditions worked really well. And all of them removed good sludge removal, 20% to 15% depending on the temperature and salinity that we used. Temperature also, the salinity also varied. Okay. So this lab data we collected, now we went back to commercial level, so whether we can do this in a farm. Okay. So we come up with the design. So this is where the farmers have this culture tank growing shrimp. We wanted to collect the wastewater coming from the bead filter put it in a holding tank, operate your sequencing batch reactor. When the ammonia nitrate level is optimum, and recirculate back. So this is our design. Very simple. Keep it simple story here. Okay. So this is the one other farming community where they use this intense farming is the raceway system, and 223,000 shrimp per meter square of this aquaculture system. Really dense shrimp. This produces a lot of sludge. Okay. So my grad student went there, set up the bioreactor in a big scale. That's Chris Lyle, my grad student. And then we operated aerobically, anaerobically the same way, 2 days, 3 days, 2 days, 1 day. That's the sequence. Then we had a control. Control is a normal reactor, no SBR. You hold your wastewater without doing this cycle. Okay. So you can see the difference. This is the SBR and this is a control. Okay. So the ammonia concentration in our SBR beautifully went down, no problem, right? So this is the aerobic anoxic mode, and we can recirculate the water without any trouble. This is nitrite NO2 level, aerobic mode, anaerobic mode, nitrate accumulated during aerobic, and then anaerobic it came down. It worked exactly what we did in a small scale. And nitrate, same story. Just accumulated under anaerobic condition, it came down. So we duplicated what we did in the lab in a bigger scale. And the total, if you accumulate all of the ammonia, nitrate, that's the data, total nitrogen. Okay. So that meets the standard of that 3.5 for ammonia, 15 for nitrate, 158 for nitrate. So the water can safely be recycled for fresh aquaculture. Then that's one success story. It could be fluke, so we had to repeat this. So we went to a different farm. This time we went to Mississippi, another state. That one was South Carolina. Okay. This one we went to Mississippi, and they have a different system. They have a greenhouse, and they have multiple tanks, and there are 2 wings. So we had to set up bioreactor one side for one wing, another side for another wing. We set up. My different graduate student did that work, and we went there. Set up this bioreactor, this SPR and a control, and they have this wastewater collection system from each wing is at the side of the building. The water comes from this, and we set up a SPR. Okay. That's how the greenhouse located in 2 different wings. We had to set up 2 different bioreactor system, raceways, and then a sediment cone, and then we set up our bioreactor next to it. Sludge on that side, we have SPR on that side. That's our design for this farm. And in this farm is a big operation, so we want to reduce the cost. So we kind of switched our process to partial denitrification. So when you look at denitrification step, it goes from nitrate to nitrite to nitrous oxide and nitrogen gas. So we can switch to partial nitrite, because when you look at the process, when you do aerobic operation, what happened to ammonia? Ammonia does the same thing, ammonia from ammonia to nitrate to nitrate. So why we had to go all the way to nitrate, right? Doesn't make sense. So what we did was, so ammonia to nitrate aerobic system, switch to anaerobic, then nitrate to nitrogen gas. You don't go to all the way to nitrate. So in that way you save some money. So that's what we did this here. So that's exactly what it is. During aerobic system, instead of going all the way to nitrate, we switched to anaerobic. So we call partial denitrification step. So we saved some money. So carbon input will be less. If you go this way, you need 100%. You need to add more molasses for denitrification. When you go from here to denitrification, you save money here. So we kind of modified the system. And there's the data for that result. The nitrate during aerobic, anaerobic mode accumulated and came down. two different sides. So there's, you see the greenhouse 1 and greenhouse 2 is what that. And then this is nitrate, same story. Anaerobically it came down. And the total ammonium nitrogen level is really worked to the level that we talked about. So water can be safely recycled. Okay. And then we did microbial analysis in this wastewater. What kind of bacteria is doing the job? So we found variety of bacteria: Paracoccus, Pseudomonas, Aeromonas, Flavobacterium. All these bacteria are dominant in our system. Okay. And then we did the gene amplification. A lot of genes responsible for different reaction. The one that dominated in our culture of bacteria is this NOS gene, which does the terminal denitrification step from nitrous oxide to nitrogen gas, which is the limiting step. Sometimes it gets stuck there. That's why your nitrate accumulate. But we didn't have any problem. We have bacteria that can do that last step to nitrogen gas. I see that genes present in all the bacteria we tested. So now we deal with the sludge. The sludge has high salt. So we need to look for the plant that can use the sludge with the salt high. So you cannot use it for agriculture because this is a lot of salt in it. So we said let's grow Spartina, which is a salt-loving plant. Spartina is needed in the U.S. for coastal restoration work because the coast is eroding. You need to go plant more salt-tolerant plant to save the coast. So we wanted to sell this to a nursery, as the nurseries can use this as a fertilizer to grow salt-loving plant like Spartina. So we did the experiment with the sludge. And this biosolids coming from our shrimp industry with the high salt, no problem, is compete with the fertilizer they use. No, they really grow well compared to control, which is they use a commercial fertilizer. And you can see they grow well, the growth of the plant statistically better. Then just the techno-economic analysis, so we kind of put this together for farmers to keep the cost down. Everything is repurposed bioreactor. We bought tanks from recycled vendors, and this is how it looks like. So we, in order to treat 2,000 liter at that concentration of ammonia, we can bring it down to 0.33, and then we can bring it down to 0.1, which is 3.55 ammonia is the cutoff. You need to bring it this level. So we can easily bring it down with the system that we design, and that's a cost. So the farmers, if they want to dispose seawater acquirement, they can save by recycling this much for one batch of aquaculture. Disposal cost we can save because they don't have to dispose, everything is recycled. The biosolid accumulation, they can sell biosolid for the nurseries. So the savings for the farmers is there. The one-time input cost to buy the SBR tank, pump, nozzles, that's going to. This is one-time investment. After that, it's maintenance. Okay, for subsequent aquaculture, they can use the same instrument. They don't have to buy it. All they have is maintenance. So look at their very first year, they're saving a couple of $100 by installing this. So keeping the system simple and economical so the farmers can benefit. So that's what we're trying to help. And this helped a couple of farmers, and as a demonstration, that's one of the successes of our project. The conclusion, we kind of showed that the SBR that we designed worked really well in the lab as well as in a big operation. It's a simple system. It's easy to operate, economical way to remediate waste. Biosolids is used for salt-tolerant plant. We have to fine-tune from farm to farm because if farm to farm temperature is varies, salinity varies. Maybe you need to fine-tune for each system. So this was funded by our Department of Agriculture for the research and the marine farming program, which is Nichols is part of that marine farming program. And my termite project was done by my grad student, when Dexter and other students helped in the project. And I was telling the story is good, but not everything was good. My student has to deal with this. When you aerate too much, we have foaming problem, right? So the aeration has to be fine-tuned during aerobic process in your sequencing batch reactor operation. The first 2 days we aerate. First day he went and switched the air and forgot to turn it off. And ended up his foamed over, came down, and he had to put a defoaming agent to bring it down. So, and then we learned the lesson. So we need to time. We put the timer. So everything is now automated. So you don't worry about forgetting to turn the air off after 2 days. Okay. So any questions? I stop here. I'll take questions. The last part was not biofuel, but I just want to show is sustainability issues dealing with wastewater system. How you can simple operation. Everything not has to be sophisticated. You can do things in simple way. To demonstrate a very simple system, so the farmers can benefit from this. So we have some application value for our research. Okay, so through science we can do things that benefit society. So that's what I'm trying to do. Thank you very much for your time.
Thank you, sir, for your wonderful lecture. Sir, one thing which I was wondering about, when you are shifting from aerobic to anaerobic process,
Yeah.
like nitrification is an aerobic process, and when you are switching from aerobic to anaerobic process, you are creating anoxic conditions.
Yeah.
So how do you maintaining those things? As a problem, you show that there was a foaming when you turn on your aerators.
Yeah.
So there was a foaming.
That was that was the beauty of the system. We were surprised that you have aerobic bacteria or anaerobic bacteria, anoxic bacteria. I thought when you operate aerobically, the anaerobic bacteria are going to die, right? We thought the system not going to work, but to our surprise, the anaerobes can take oxygen, but they go dormant. They are not, active. And then when you turn the air off, the aerobic bacteria, some of them may die, but they are not active. Then anaerobe, take part in the biochemical reaction and carry out the next step. So that's what we thought. We thought we're going to lose these bacteria because the conditions are so different. But bacteria are so robust and they adapt these conditions so quickly. And when we take sample and during aerobic system and culture them anaerobically, they grow. They didn't die. So that means the bacteria are surviving, but they are not active. That's what the difference is.
One more thing, sir.
Yeah.
You mentioned about partial nitrification.
Partial denitrification.
Partial denitrification. Have you tried with some Anamox bacteria, like in which we are?
Yeah, Anamox is very, very, very slow growing bacteria.
Yes.
Very slow. The farmers cannot wait. So yeah, Anamox is good. Academically and in the ocean system, Anamox is good. In ocean, that's what the nitrogen cycle is, and Anamox play a good role. But in our 1 week operation, it didn't grow. We did Anamox, but Anamox take 4 weeks, 5 weeks to get the critical mass to do the nitrogen cycle.
Yes, sir. Just because their yield is very less, maybe 0.1 gram Vs per.
Yeah, and then it doubling time is
very
in
less.
weeks, not hours like other bacteria.
And sir, any specific reason about selecting molasses during?
Molasses is the cheap one because molasses, if you look at any kind of carbon source available in the US, cost-wise, molasses is the cheap. You can get molasses 5 cents per gallon, right, compared to any other carbon source. So.
Maybe a glycerol or recirculated sludge, maybe.
Which one?
Recirculated sludge, maybe a good carbon source during denitrification.
Yeah, we can try any other carbon source, but we prefer molasses because the reasons it's available, plentiful, because we have a lot of sugar factories, and also it's very cheap. That's the reason we chose molasses. Yeah.
Okay, so means in that way molasses is also managed, and again thank you, sir.
Yeah, molasses is also there. The farmers we talked about yesterday about biorefineries, so they can sell to the aquaculture people, another route to sell their product. Yeah.
Definitely, sir.
So anybody? No questions? Okay. Anybody else?
Sir,
Yeah.
one more follow-up question, sir.
Yeah.
Sir, you mentioned about functional similarity and with no statistical difference during during your studies of termite gut.
Yeah.
Sir, anywhere you found out the environmental stress may be the region of the adaptation when you were switching between the species?
Yeah, when you talk about termite microbiome, what we found was diet make a difference. The species difference is, there is no difference in species at all, whether termite come from different faraway places eating different woods, but what is the difference is the abundance of each species. That's what we are finding the major difference. Again, that you could call the stress, you could call adaptation, you could call anything, but that's what we are seeing. We are seeing the bacteria and protozoa are the same, the species wise, but the percentage of each one of them varies from type of wood they eat. Okay? So you think of this in terms of feedstock. In the one we talked about yesterday, variation in feedstock. Okay? The termite gut microbe is adapting to the feedstock. Yeah.
And one more unique thing which you talked about was the selective uptake of phenol.
Yeah.
Maybe due to repression, or you call it a gene elimination. Sir, just wanted to know more whether it was a repression or it was a complete elimination of gene or
the phenol.
Sir, catabolite repression which you were mentioning.
Yeah.
Selective utilization of phenol over glucose.
Yeah.
So the sole reason for that, sir,
could be a repressor, and if you put the bacteria back into glucose over a long period of time, it may come back. You're right. The phenol is a repressor.
Yes.
For glucose metabolism. Yeah.
Right.
Yeah. Exactly. Yeah. Any other questions?
Any question from the online candidate?
Okay. No. Okay. Thank you.
Thank you, sir. Yes, sir.
You have all the PowerPoints. You can for them to.
Yes, sir.
You can
We'll
distribute
share it.
everything to everybody.
Yes, sir.
You can. Yeah.
After the break, we'll check it.